Hello, my name's Dr. Sue Whittle and I'm a senior lecturer at the University of Leeds, reading biochemistry. Today we're going to look at how we can monitor the activity of enzymes and how we can find out about the way in which substrate concentration, pH and temperature affect reaction rates. Enzymes are proteins and they act as biological catalysts. In other words, they increase the rate of biochemical reactions by decreasing the activation energy of the reaction, i.e. the amount of energy that's required to allow the reaction to occur. Enzymes convert their substrates into products, sometimes with the help of additional molecules known as coenzymes or sometimes cofactors. We often use the term enzyme assays to describe experiments where we're looking at enzyme reactions. In principle then, in order to monitor the rate of an enzyme reaction, we need to be able to track either the appearance of a product of the reaction or the disappearance of a substrate. There are lots of possible techniques for doing this. As one example, we could use a radioactively labeled substrate, and then we could calculate the amount of radioactive product that we've got formed. Sometimes a reaction produces a gas that we can measure. You may have used that technique at school to measure the rate of a reaction of an enzyme like catalase, which produces oxygen. One of the commonest methods for assaying an enzyme is use of spectrophotometry. This relies on the presence of a substrate or a product which absorbs light. And I'm going to demonstrate a couple of different enzyme reactions today using a spectrophotometer. You may use a colorimeter at school. This works on a similar principle. Basically, spectrophotometry works by determining the amount of light at a specific wavelength that is absorbed by a solution. We know that solutions look coloured because they absorb light. For example, a blue solution looks blue because it absorbs all the other wavelengths of light. So it's only the blue light that passes through the solution to our eyes, and so we see the solution looking blue. Using this idea, the colour of a solution will often give us a clue about which wavelengths of light the molecules in that solution actually absorb. By using a wavelength of light which molecules in solution absorb, we can monitor changes in concentration, because the amount of light that is absorbed is directly proportional to the concentration of the light-absorbing molecule within the solution. This is known as Beer's law. The first enzyme that we're going to look at is a phosphatase, and this catalyzes the removal of phosphate groups. We will look at the structure of a phosphate group in a moment. Phosphate groups are found on many types of molecules in living systems. You may already be aware of nucleotide phosphates such as ATP. These are important in energy transfer and, of course, in the synthesis of DNA and RNA. But many proteins can also have phosphate groups added to them, and this is often an important form of biological control, for example, controlling the rate or the activity of an enzyme. Not surprisingly then, there are a number of different phosphatases found in living organisms. These enzymes are quite unusual in that they often use quite a range of different substrates. As I'm sure you're aware, most enzymes are highly specific and utilize only one, or if you're lucky, a very small number of closely related substrates. But phosphatases tend to be quite non-specific. In the lab, we can use this property to our advantage. Phosphate does not absorb light, so at first sight, spectrophotometry doesn't look like a suitable way to monitor phosphatase activity. However, because these phosphatase enzymes will remove phosphate groups from lots of different substrates, we can use what's known as a chromogenic substrate. And that's an artificial substrate which, when the phosphatase removes the phosphate group, produces a coloured product. On this slide, we can see the reaction that we're going to use. The substrate is 4-nitrophenyl phosphate, And when the phosphate is removed by the enzyme, the product formed is 4-nitrophenol, which absorbs light at the wavelength of 405 nanometers and looks yellow. You can see the phosphate group on the left-hand side of the equation attached to the 4-nitrophenol. And on the right-hand side, you can see it as a free ion. As you know, enzymes are affected by pH, how acid or alkaline the surrounding medium is. Each enzyme has a pH optimum, the pH at which they work best. This can be highly variable. For example, lots of enzymes in cells have a pH optimum close to pH 7, because this is the pH that most cell cytoplasm actually operates at. On the other hand, pepsin, an enzyme which works in the acidic environment of the stomach, has a pH optimum at around pH 1.5. Phosphatases come in two families: acid phosphatases, which have pH optima around pH 4, and alkaline phosphatases, which have pH optima around pH 9 to 10. In the experiment we're going to do, we're going to find out what sort of phosphatase we have and determine its pH optimum. Here I have a number of reaction mixtures set up at different pH values. They all contain buffer to keep the mixture at the required pH, and the substrate for nitrophenol phosphate. You'll see that for each pH I'm going to investigate, I have 3 tubes. 2 of these are duplicate reactions, which will increase the accuracy of my experiment. And the third one is a control, which will not contain enzyme. The control is to make sure that any color change observed is due to the action of the enzyme and not to anything else. For example, the substrate degrading on its own at particular pHs. I have added water to replace the enzyme in the control tubes. I already have several sets of reaction tubes incubating in the heat block, to which I've already added the enzyme. I'm now adding my enzyme to each tube, mixing the contents and putting them into a heat block to ensure that the temperature remains constant throughout the experiment. After 5 minutes, I will stop the reaction by adding a very strong alkali, sodium hydroxide, which will denature the protein and prevent the enzyme from working any longer. This type of experiment is called a discontinuous assay because I'm not monitoring the reaction as it goes along, but choosing a particular time point to measure at. And from this, I will calculate the reaction rate. So now the reaction is complete, so I can measure the absorbance of each tube. To do this, I will pour the contents of my reaction mixture into a special tube called a cuvette, which fits into a holder inside the spectrophotometer. If we look inside the machine, we can see the cuvette holder and the lens that the light comes through. The light is passing in this direction through the solution. As I want to compare the color formed in the presence and absence of enzyme, I am going to set the machine to 405 nanometers and then use my control tube, the one without any enzyme, to set a 0 value. Basically, I'm setting the spectrophotometer up to detect only the difference caused by the enzyme. It will ignore any absorbance caused by the unchanged substrate, the buffer, or the plastic sides of the cuvette. I can now measure the absorbance of each tube. You can see that the intensity of color varies at different pH values. I can then take an average of my duplicate tubes and divide that value by 5 to get a reaction rate per minute. I'm making a major assumption in calculating the reaction rate this way. You might like to discuss what this assumption is and how you could test whether it's a fair assumption for me to make. It's really important when designing an experiment that we think about whether we are making assumptions and whether we can justify making them. Now that I have my results, I can plot a graph of reaction rate against pH, and here it is. From this we can see that the phosphatase I have been given is an alkaline phosphatase, and it has a pH optimum at pH 10. To understand the effect of pH on the rate of the reaction of enzymes, we need to remember what enzymes are. They are proteins which consist of one or more chains of amino acids held together by covalent peptide bonds. The three-dimensional shape of a protein, however, is determined by a series of weak interactions between the atoms involved in the peptide bond and between the amino acid side chains. At pH values far from the pH optimum, the very high or very low pH disrupts these weak interactions, causing the protein to lose its specific three-dimensional shape. In other words, the protein is denatured. At pH values closer to the optimum pH, pH can affect the reaction in a variety of different ways. By affecting the charge on the amino acid side chains in the active site, for example, or by altering the state of the substrate. Therefore, you get this bell-shaped graph. If I repeat the same experiment using a number of different heating blocks set at different temperatures, I can also see the effect of changing temperature when I keep the pH the same. Here, we see a slightly different shaped graph. As the temperature rises, the rate of reaction increases. This is for two reasons. Firstly, the warmer the temperature of the solution, the more energies that the molecules of the substrate have, and the more actively they move around. This increases the chance of an enzyme molecule colliding with substrate molecules. The substrates have to enter the active site for the reaction to occur, and the more rapidly everything is moving about within the solution, the more collisions there are likely to be.